So the next talk is Nitya Singh from University of Florida. It's going to be a pre-recorded talk, but Nitya is on site to to answer the questions afterwards.
Hello everyone. The topic for my talk today is modeling the Campylobacter infection dynamics and estimating the immune parameters for the malady study data, and by applying the approximate Bayesian computation. So as we all know that the enteric infections, especially by the Campylobacter species, they are quite a burden especially on the young children, and they are implicated in reducing their growth and also for stunting. So all the preventive interventions, they basically rely on the efficiency of the exposure source attribution, but most of them, they only use the proportion of symptomatic cases or the population attribution factors while ignoring the dynamics of acquired immunity and the asymptomatic infections. It has been shown previously by Havilar et al. That the boosting and the waning immunity phenomena, which is very crucial to estimate the incidence for these diseases. And they have proposed a very simple model which involves the 4-compartmental model where the population from the susceptible state can move to the protected state with a probability of illness pi and a force of infection lambda. But this transition either can be asymptomatic or can be symptomatic. People can be ill or people cannot be ill. But in this state, protected state, they cannot get infected further or get ill. But due to the waning of the immunity, they move to the compartment they called partially protected compartment. And from here, with the further waning, they can move back to the susceptible compartment. But an interesting phenomena happens between the protected asymptomatic state and the partially protected state because of the high exposure, that there's a loop between these two compartments that with the rate of alpha lambda, that the person moves between these two compartments and never get ill. And so these are the important considerations for estimation of the incidences. So that we see here, they propose by the numerical example that in case of low exposure shown in these two bars, that only the susceptible people who are at the risk of the disease, they should be considered in the cohort or the case control study. But because of this immunity, even at the high dose, a fraction get partially protected or asymptomatically protected. But in the case of high exposure areas where there's a maximum of the population, it is protected either asymptomatically or partially. So there's actually in the population, there are a very small fraction of the people who are actually susceptible. So when the estimates are made, considering the whole population, which is not at the risk, they are highly biased. But the major hurdle was in applying this model in estimating the real estimates was the lack of the experimental data. And there was only one challenge studies so far. But then came the malady data, which was particularly useful for our purposes and for this model, which was collected across 8 countries, which had suspected high force of infection. And that is why we thought that study would be particularly suitable for estimating the immune parameters. And interestingly, that all the data was collected starting from the age 0, and there were regular follow-ups till the age of 2. And the data was collected in a manner that it could differentiate between infection and the illness. But because of the different patterns in the immunity of child during first and the second year of the age, we could only use the first year data and only across the 5 countries because of the not enough data points. But we had enough, as 1,100 children, they completed the follow-up. And we have almost 3,800 episodes of diarrhea and 11,500 asymptomatic stools, which were tested both for Campylobacter species by ELISA and for Campylobacter jejuni coli by PCR. When we fitted this data to our model compartments, we found what we were expecting, that there was quite a fraction of the population was asymptomatically protected. You can see this by blue graphs, blue dots, and there are the confidence intervals. Also, there's a difference in the pattern in the Campylobacter species and jejuni and across all the countries, but the trend is same. And if you see at a little high resolution, the fraction of actually ill people who get protected with the symptomatic illness, that there's a trend different in different countries, but the fraction is very low comparison to this the whole protected population, asymptomatically protected. And also interestingly, because we don't have any method which can tell about the immunity status of partially protected, so we could not separate the susceptible population from the partially protected population, and these two are together. Because of this reason, not any maximum likelihood method could converge, and because the likelihood was quite flat. And we also tested estimating using the Bayesian methods, which were quite unreliable because what we were seeing was almost the same as the prior. So none of those methods worked for estimating the parameters for this model using this data. So then we used this ABC simulation method, and which is a distribution-free numerical method, and starts the simulation using the Bayesian priors. But at each cycle, it optimizes the error. And you can keep optimizing up to the threshold of the error you want to optimize, like as much as find like 0.0001 error. But the little bit of limitation here that the computations are really expensive, and we could do that by using the UF Hypergator facility, which is a supercomputer world class in USA. But and we tested almost 160 combinations using a synthetic data which we generated like MALED data, and for the 3 waiting time, prevalence of diarrhea and asymptomatic infections. And we got quite reliable results, and used from these estimates, we back estimated the parameters. And you can see that these are the priors, these black lines, and this, it converged with each cycle, it converged to a sharp peak. So the 95% confidence bound intervals were quite reliable. So then after testing and validating, we used this method for the real estimates of MALED data, and we found that Jejuni coli, they have a force of infection lambda for in the range of 3 to 7 years in these countries, and species in the range of 5 to 20 years, which is quite high force of infection. And you can also see that this the duration of full protection, which is inverse of alpha, this is in the order of this days, and the duration of partial protection, which is the inverse of gamma, is in the order of weeks. But because this force of infection is quite high, this can affect the epidemic quite considerably. Using these estimates, although these are preliminary, we also move ahead and estimated the incidences, which are also quite high in comparison to the MALED reported incidences. So, but these estimates are under still cross-validation because there are multiple possible solutions, and also you have find a trade-off between the error threshold. But in summary, I can say that these cohort study, they provide an excellent source of data for estimating these immune parameters for these such dynamic disease models. And these models also allow a novel method to quantify the pathogenic specific diarrheal incidences, and they can also be applied to estimate the impact of changes in the exposure frequency and dose. But there is a limitation that there is no data available to confirm the immune status of the population, and we need such methods so that we can calculate the illness incidences very accurately for both symptomatic and asymptomatic infections. Thank you. There are some references, and we would like to acknowledge RIVM for their support. And I would like to take any questions. Thanks.
Thank you very much. Are you able to? Oh, you're able to join us. Thank you. So, so we've got a question. So I might have missed this as well, but when you're talking about the samples for the coli and the jejuni, it wasn't clear to me from the stool samples. Was there a culture step involved? So has there sort of been a selection for coli and jejuni within there?
I guess yes, because that's the detailed protocol is available in the MALED published paper, and what we used for our purposes was the data which they published, that they sampled, and using all the protocols they standardized and they tested by ELISA, which we are assuming that is the test for the whole Campylobacter species. And they tested by PCR selectively for jejuni and coli. So definitely there was a selection process for jejuni and coli because they tested and confirmed and reported that it is for jejuni and coli. That is in the published study when they published this data. Yeah. So it is coming from there.
Thank you. Okay, I think you've just answered that within your answer. Okay, I think that's all we have. So thank you very much.
Okay.